So today we'll be discussing about the biosynthesis of purines, all right? So whenever we say purines, what are we talking about? We're basically talking about what? Two compounds, right? And that is here, let me write down here, adenine, all right? This is an adenine, all right? And then other structure basically a guanine, all right? This is what we're talking about. And I'm showing you the two structures right here. The nitrogenous base, right? This is adenine, right? And this is guanine. And why are we studying this? Remember, these are purine structures, right? And in our DNA structure video, I talked about it, okay? That why we use purine, or why do we study purine, right? The most important thing that you have to remember is that purines are used to actually what? Synthesize DNA and RNA. That's one of the main functions of purines, meaning that you use this adenine and guanine to make what? DNA and RNA, right? That's something that you have to remember. Because what is DNA and what are RNA? These are basically what? The polychains, right? These are basically what? Polychains of this, nucleotides, right? And when we say nucleotides, what are we talking about here? Nucleotides, when we say this, we're basically saying that you have a purine base, which is one of this, right? Purine base, right? Your purine base or nitrogenous base, plus you add sugar, all right? Which is a ribose sugar, plus you add a phosphate, right? And this structure is called what? Nucleotide, right? So the reason why we have to synthesize we have to basically make this adenine and guanine, these structures, is to make DNA and RNA. That's one of the functions of this, right? Now, not only this, right? Not only this, you have to remember this, the whole idea of you need this adenine and guanine, which are basically what? The nitrogenous base, right? You can find this in what? Your diet, right? And you can take it. But most of the times you actually endogenously inside the body, you actually make this compound, right? And look, these purines, and not only purines, the pyrimidines are there. They are used obviously for synthesized DNA and RNA, but they also use what? Not only the functions of this, but they also what? Ultimately, what they also make something called ATP, right? So there is a what? High energy molecules, right? We use this to make what? High energy molecule, right? High energy molecules. Not only this, if you remember, there is something called cyclic AMP, right? There's something called cyclic GMP. These are what? These are basically second messengers, all right? These are second messengers. And these, what is this called? Adenine monophosphate, guanine monophosphate. See, we're using this adenine and guanine, for basically regulating. They basically regulate for the metabolic regulators, right? For the cells. Not only this, we use this. Like for example, other areas, like for example, if I want to mention this AMP, just AMP, right? Or just GMP, what do? They can also use for the different enzymes, right? They can allosterically regulate different enzymes to do their functions, all right? So these are basically general functions of why you need adenine and guanine, all right? Now, then after you have to remember this, okay, where do you find this this adenine and guanine? As I said, it's a nucleic acid. You can actually take it from the diet. But also one more thing you have to remember here is that this small amount you can take it in the form of diet, but most of the time we actually make it in our body, right? And why do basically this purine synthesis actually takes place? That is something that you remember. The purine synthesis takes place mostly in where? In the liver. So you can think about this as whole as a liver, okay? The synthesis will be taking place I'll just write down here, synthesis will take place in liver, right? This is also sometimes you have to remember. Now, remember, we're talking about the purines, but there's also pyrimidines we'll talk about in next lecture, right? Those are also required to make what? The DNA and RNA. And those pyrimidines are basically what? Those are usually made by cytosine, right? Uracils and basically thymine, right? But we'll talk about this in next lecture. But now, today we'll be doing is that we're actually synthesizing this structure right here, okay? This adenine, how do we synthesize this structure? This is what we'll be talking about. This is guanine, all right? So basically we have to make this structure together. Now, first we'll start with the purine synthesis, right? So we're synthesizing this purine. So to do that, all right, you have to remember this something. In the liver, right, we take basically, there are GLUT2 transfers are there, right? And you basically take what? You basically take a 6 sugar molecules. So you can simply say this is a 1 carbon, this is a 2nd carbon, right here, 3rd carbon, right? And the 4th carbon right here, 5 carbon right here, and you have 6 carbon right here, right? I'm basically saying this is your glucose structure, right? Your glucose, so the GLUT2 transporter, you basically take in the liver, right? And then what happens, once it's in the liver, this can actually go and basically make something called glucose 6-phosphate, right? Glucose 6-phosphate, because there are enzymes, like for example, glucokinase, hexokinase, and all those enzymes, right? Now, ultimate result is this, you go this pathway, and very importantly, right, you actually use this pathway called pentose, all right? Pentose phosphate pathway, all right? Or you can simply say this is a hexose monophosphate shunt, okay? Same thing. Using this pathway, ultimate result is actually you'll go and make compound called ribose, all right? Ribose, this is a ribose 5-phosphate, all right? This is the compound you'll make. Okay, I'll erase this and then I'll just write it down here, all right? Ribose 5-phosphate, this is the compound you'll make, all right? So basically, with the glucose and with the help of basically hexomonophosphate shunt pathway, you'll make a ribose 5-phosphate. I already discussed about hexose monophosphate shunt in a different video. If you want, you can go and watch that lecture. But for you, in order to actually synthesize purine, what I want you to remember is that the initial step is you need a ribose 5-phosphate. This is a sugar, right? It's obviously, because it's coming from sugar, right? So now the point being here is that, okay, how do you, what is ribose 5-phosphate looks like, right? So we'll draw the structures of this, right? So remember, you basically have what? You have oxygens, and it is a 5 molecules basically sugar, right? It basically looks something like this, right? And then we can simply say, all right, because we'll label this too. This is a carbon number 1, okay, 1 prime right here. 1, this is 2, this is 3, this is 4, and this is 5 right here. And at the carbon number 5 positions, what do you have? You have basically phosphate group is attached to it, all right? So basically what happens, you have oxygen right here, and this is what? PO4 3 minus. So you have at carbon number 5 prime positions, phosphate is attached, right? This compound, look, 5, right? This is a ribose sugar, right? Ribose, and the 5 position, there's a phosphate. That's why we call them a ribose 5-phosphate. This is a ribose 5-phosphate, right? Now, where you are using the structure, right? And obviously, look, you can have, right here, look. What is a compound? You have a hydroxyl group here. Okay, you have a hydroxyl group here, and basically you also have a hydroxyl group, and you have a H here. It looks something like this, right? Now, because remember we said, in order to make nucleotide, right? That's what we're trying to do here, right? What happens is that, you need purine base, you need sugar and phosphate. We have the structure, right? Then we need to make a purine base, right? Then we can make a nucleotide, right? So this is what we're talking about. We're trying to make this purine base right here, right? And how do we make this structure right here? That's what we're trying to do. Because ultimately what we're going to do is we're going to make this structure. That's what we're going to do here, right? Now, this is a ribose 5-phosphate, right? Now from here, what you have to remember is that there is important enzymes that you're going to see right here, all right? This enzyme is going to do what? This guy is going to take, there's an enzyme called, all right, I'm going to write down here. This is called phospho, all right? I'm going to write down big, okay, phosphoribosylse. This is called phosphoribosylse pyrophosphate. All right, pyrophosphate synthetase enzyme is called. All right, synthetase enzymes. And sometimes they write this as a P, phosphoribosylse with PR, all right, PP, phosphoribosylse pyrophosphate, all right? This is how they write down. What they do is, look, phosphoribosylse means there is a phosphor and there's a phosphate here, there's a ribosylse here. We draw the same structure right here. It's nothing crazy. You basically make a same structure right here, right, like this. Same structure, nothing changed here, all right? And I'm just gonna write this phosphate, I'll just write down P, okay? So that way we save the time. But you got the idea. Now, you have this phosphate and ribosylse. So that's why we call them a phosphoribosylse, right? But what is pyrophosphate basically means that. All right, look, you remember this. All right, I'm gonna write just hydroxy in the blue color here. Hydroxy here, hydroxy here. Here, basically in here, look, this position, carbon number 1 position right here, you're basically adding a what? Phosphate, 2-phosphate but they're more in a condensed form, right? Pyrophosphate referring as that, they basically have astral linkage into it, right? That's why we say pyrophosphate. Now, if you look at this carbon number 1 positions, this enzyme is basically going to do is that you have oxygen here, right? And you have a P right here. Look at this. You have a P right here, oxygen P right here, and then you have you just write down a phosphate, right? 2-phosphate double. You can say this is a double bond right here, right? And then you can simply say this is O right here, right? And then you have, what is this? After that O, then you basically have P, right? Double bond right here. And you can say O or O minus, like this. Look at this. Look, this is basically what? Astral linkage right here, right? So this is a pyrophosphate, right? That's why, sometimes, there's a different names are given for this, right? So sometimes they also call this, okay, there's a ribose here, right? You have sometimes, okay, I'll write down here. You have, this is ribose, okay? Ribose, this is ribose. What? Phosphate, okay, ribose, phosphate, right? Coming from, but then you basically adding a 2-phosphate group into it, right? Here, in the form of ester. So sometimes they also call it diphosphokinase, all right? So this is a ribophosphate diphosphokinase. Sometimes they call this, or sometimes, they make things crazy here. Sometimes they also say this, okay, there's a phosphoribosylate, okay, you can say phosphoribosylate, right, to be more accurate. Phosphoribosylate diphosphokinase. Actually, let me write down here. This is something they call phospho, all right, let's say ribosylate, right, diphosphokinase, or sometimes they call it phosphoribosylate pyrophosphokinases. Instead of saying synthase, they will say phosphoribosylate pyrophosphate kinase. That's also another name, all right? Basically mean the reason why they call kinase is because what does kinase mean? You're basically adding a phosphate group, right? But for the step 1, a lot of times, they use this enzyme, phosphoribosylate pyrophosphate synthetase enzymes, or PRPP, right? But I'm just giving you, if you see, this other enzyme, don't get confused. Just read it. Phosphoribosylate and diphosphokinase, the same thing, right? You're just adding a phosphate group into it. Now, this is what happens. Look, now this guy, right? This one, when you do this, right? You basically got what? In a carbon number 5, you have what? Phosphate group, right? You have a phosphate group there. And this compound is what? Basically, phosphoribosylate pyrophosphate. So this is, okay, I'll just write down here. I'll just write down this is phospho, all right? This is phosphoribosylate, right? Ribosylate, pyrophosphate, right? This compound is this. So, or simply you can write this is a PRPP, right? This is what you're making. Now, after this, what do we do? There's another step that will go, all right? Another step. And this is, look, and guys, you don't have to remember the structure if you don't want to, right? But I'm actually, I'm actually showing you how things happen. So, rather than just quickly memorizing it, all right? This is for understanding, all right? So now, let's make here another enzyme. And by the way, guys, to be honest with you, this phosphate, like this 2-phosphate, where did they come from? This enzymes, all right? Let me delete this, okay, now. Erase this. You got the idea. What happens is that, they actually use this enzyme. There's actually this thing, cofactor. They use basically ATP, okay? This guy takes 2-phosphate group from the ATP and makes it what? This is AMP. That's what they do. And also there's a magnesium is also there. It's very important, right? So basically, it basically takes 2-phosphate group and puts it here, right? And when you do that, what do you make? You make AMP, all right? This is something you have to remember. Now, another thing here. Now what happens again? We're going to make exactly the same compound right here, all right? That is a ribose sugar. This is ribose sugar here, right? This is a ribose sugar right here. And we're making this is a phosphate group potassium, right? Now, this is hydroxyl group here. This is hydroxyl group here, right? Now, what is happening here is that, look, that, there's a special enzyme that you're going to see here. And this is a very, very important enzyme that you have to remember, okay? This enzyme is called, all right? This is called, okay, we'll just write it down. I'll write down in red, so that way you remember this enzyme. This is called glutamine, all right? This is called glutamine, all right? Glutamine, right? But it's amido transference enzyme. This is like a rate limiting step. This is like a rate limiting substance. This is like a rate limiting step of the purine synthesis. The glutamine amido transference enzyme is there, right? And this is what this is going to do. And why do we call them a glutamine is because, look, it uses, all right? It basically uses glutamine, all right? Glutamine and convert into glutamate, right? And now for that one, I'm going to draw and show you how glutamine looks like, all right? Look, you have a carbon double bond with oxygen, and you basically have hydroxyl group into this, right? Okay, I'll make it right here. So you know this is what I'm talking about. I'll make it in a, let's just say black color, and I'll use this, okay? So you have basically carbon double bond with oxygen, and you have a hydroxyl group attached to it, right? And then you basically have, look, amine group attached to it, right? And there's a glutamine we're talking about, right? You basically have a compound like this, and you have a carbon double bond with oxygen, and you have a, look, you have a H2N. Look at this. This is the structure of glutamine, all right? This is a glutamine. Now, this glutamine, they use this glutamine, all right? They use this glutamine, so we'll make this as this structure is a glutamine, all right? So glutamine. And when they use the glutamine, what does this structure become? It becomes glutamate, right? Glutamate, it will become. So basically, look, this glutamine, look at this. You see the nitrogen right here? Glutamine is this way, right? This is a glutamine structure, right? And they are basically transferring amide group, right? And putting in here. That's what they're doing, right? And doing that, right? Well, doing that, what happens? This structure will become basically what? A carbon double bond with oxygen with a hydroxyl group, right? And then you have NH2 group here, right here. Same compound, look. There's nothing more carbon double bond, but this will be hydroxyl group here. And that will be glutamate, okay? This is a glutamate structure now. And when doing that, there is also release of PPI, meaning that they lose this pyrophosphate, all right? Now, then what is that? Then look, this is a glutamine, and they are used, and then the other question you have to ask, what is amide referring to? Amide is nothing. This is what amide is. Look, if you have a carbon double bond with oxygen, right, and you have a nitrogen base, which is attached to some sort of R group, like carbon compounds, right? There's R prime, and maybe this is another R, and you have basically R prime. Look, this is structure. This is called amide, and that's what exactly happening down here, right? That's what exactly happening here. Now, when they do this, right, they basically attach onto this position, on the same carbon position, and they do basically on this position right here. What is this? This will be your amine group, like I'll just write down a H2N, right? So when you do this, right, you attach H2N, and for that one, right, what do you call this? You basically call the structures, all right, phosphoribosyl. Look, this is a phosphor, this is phosphate, this is what ribose. So we'll just write down phosphoribosyl, right? Phosphoribosyl, and you adding this group here, amine, right? Phosphoribosyl amine. That's what it's called. Because this is a phospho, and you're adding, there's already a phosphoribosyl, but you're adding amine group because of the amide transference, right? And you basically make a compound phosphoribosyl amine, right? That's how you make it. Now, the question is that, if you want to be very, very accurate, look at the conformation, their positions up. This is actually facing upward, right? And this one is facing downward, right? Because ultimately, you see the hydroxyl group right here? Based on locations of this, there's something called alpha and beta. Like, for example, if they're facing like downward like this, like based on the hydroxyl positions, like this way, this carbon is facing downward, right? Where the hydroxyl group is, we can call them as alpha, right? Alpha. And this one you can call a beta. But, just for now, for this step, they're not going to ask you that. What is more important is that, just remember there's a phosphoribosyl amines are formed based on, because of this enzyme, glutamine amide transference enzyme, right? And this glutamine is basically coming from the amino acids. That is very, very important, right? And I also try to show you how glutamine looks like, right? There's a glutamine compound. And glutamine is what? It's an amino acid. It's a polar amino acid, right? Because it has a charge there. Now, from the phosphoribosyl amine, right, what are we going to do? Okay, we're going to make down here. Okay, I'll just quickly draw the phosphoribosyl amine, right? This compound right here, phosphoribosyl amine, this is NH2 right here, right? And you have a P group attached to it. And you have a hydroxyl here, you have a hydroxyl here, right? So this compound is basically phosphoribosyl amine. Now, what is going to happen to the phosphoribosyl amine? Now, again, it's going to be important concept here. Look, the ultimate rule here from here is that, look, what we're trying to do, we're trying to make like adenosine monophosphate, AMP, and then we're trying to make guanine monophosphate, right? That's what we're trying to make ultimately. What happens is that, there's a lot of steps that's going to go, all right? It's going to be a lot of, it's about like 8 steps it's going to go, right? About 8 steps, right? And what happens is, ultimately what happens is that, they're going to make a compound called inosine monophosphate, okay? IMP. This is what they're going to make, this inosine monophosphate. Okay, I'll write down a big. Ultimate result is they're going to make something called inosine monophosphate. All right, that's the compound they're going to ultimately make. Ultimately, this is the compound they're going to make. All right. Now the question is that, do you have to remember the 8 steps? There's all this. No, because it's very complicated, and I myself do not know it either. So just know that there's a lot of stuff that goes, and the ultimate result is that they'll make inosine monophosphate, and this inosine monophosphate is the first nucleotide. This is an intermediate. Okay, this is an intermediate. Now that we have talked about what is the structure that happened in this X step. Basically, what they did is that they actually tried to make this structure. In those S steps, they'll form a structure like this, okay? But what is that structure? We have to remember this. Now I'll draw right here, okay? Now I'll make it in a red so that way you'll know different. So basically they'll make, after that, it will be like this. You basically have this, look at this. What is this? This is your one ring. Then you have another ring like this, nitrogen, right? And then you have a double bond like this, correct? And you have another N compound right here, obviously the H. And then you have this structure right here. You have a double bond here, all right? Double bond here. That's what happens. And then you also have this compound, because it's going to have a HN right here, and you have a double bond right here. Double bond, and you have an N compound here like this, okay? And here, at this position, you have an oxygen group here. Look, this is, I'm going to label 1, 2, 3, 4, 5, 6, right? 7, 8, 9. All right? Now, the point being here is this. Okay, let me erase this and I'll explain to you. All right? Look. In those S steps, right? What is happening here is this. In those S steps, what happens is there are enzymes, like for example, not enzymes, amino acids. Like for example, glycine. All right? Glycine is very, very important. Okay? Glycine, glutamine, aspartate, all right, aspartate, tetrahydrofolate, or also called BH4, right, and carbon dioxide. This guy is going to play a very, very important role, right, because forming these structures, all right. So those are the different steps that will take place, right, where basically these glycines, glutamine, aspartate, THF, tetrahydrofolate, carbon dioxide, they play a role in forming these structures. Right, this structure that we just did. Now, right? So now that means that we have to know that, okay, quickly, let's just say, I'm going to draw what glycine looks like. For example here, carbon double bond with oxygen, right? And you basically have, what is this? You have a hydroxyl group here, and you have this functional group right here, right? And you basically have H and H. Like, look, this compound is glycine, okay? Now this is a glycine. Look, you see the glycine has an amine group, nitrogen group into it, right? And there's a carbon group here too, right? Other structure, let me make. Okay, glutamine we already drew, but again, let me just quickly draw for you. Carbon dioxide, oxygen, right? You have a amine group attached to it. And you basically have, what is the structure? Carbon double bond with H2N. That's a glutamine, right? Let's draw another compound, which is aspartate, right? Which is a carbon double bond with oxygen with a hydroxyl group right here. And you have an amine group attached to it like this. And you have, look, one structure, and you have this structure, and you have a carbon double bond with oxygen, right? And you basically have a amine group, okay, if it's aspartate, right? You basically have, look, hydroxyl group there. So think about this, right? Look, these are a different type of structure. You have glycine, you have this is glutamine. Okay, let's just say this is your glycine, all right? This is your glycine compound, all right? This is your glutamine, right? Glutamine, and this is your, what is this? Aspartate, right? This is your aspartate. Now, look. The point being here is that, during this process, this nitrogen, okay, this carbon and this carbon, right? This carbon is basically, I'm going to make it, all right? I'm going to make it blue. This carbon, this carbons, and this carbon right here, right? These are actually, synthesized by glycine, okay? Blue is glycine coming from there. That's what's going to happen. Now, then after that, you have to remember other stuff. Then what about, look, then we have to see, what about this carbon right here? All right, this carbon, right? This carbon right here, right? This carbon, which is basically on the 8th position, right? This carbon is coming from THF. The donor, okay? Donor of this carbon is a THF. Same thing happens on this one. On the carbon number 2, this carbon right here, the donor is what? THF, all right? Tetrahydrofolate. Now, then you have to remember, the other compounds are there too, which is this one. this nitrogen, right here, this one, carbon number 3 and carbon number 7, right? These guys, right, they're coming from basically glutamine, all right, glutamine. Now, we have a 1 carbon and then 1 nitrogen. This nitrogen right here, right, this is coming from aspartate, right, this aspartate. And carbon down here, this is coming from carbon dioxide, right? See, this is how, right, you make a compound. You make this compound, right? But remember, this compound, right, what is this compound though? This compound, right, with this, all this stuff, glycine, glutamine, aspartate, THF, right, BH4 and carbon dioxide, right, we use this to synthesize this compound, right? But this compound is not an adenine or it's not a guanine, okay? This compound is none of those. Then what is this compound? Because in order to make an adenine, okay, what do you need to have on this position? You need to have this, right, amine group, right? There's no amine group there, correct? Because it's an adenine. And if you want to be guanine, it kind of looks like a guanine in this compound. But look at this. Label this. 1, 2, 3, 4, 5, 6, 7, 8, 9, right? Same thing. 1, 2, 3, 4. We're just labeling this carbon structures. 6, 7, 8, 9. It kind of looks like guanine, but look at the carbon number 2 positions here. There's nothing there, right? There's nothing coming out from there, right? So that's why this compound, it's not an adenine and not a guanine. Then what is this compound? This compound is hypoxanthine. What is this compound? Hypoxanthine, all right? This is a hypoxanthine. This is a hypoxanthine. Now, right, hypoxanthine. But the most important thing is this, that, you know, all this carbon, right? This carbon is right here. The most important thing is this. You need the 2 ring structure, right? The 2 main ring structures, those are similar in hypoxanthine, adenine, guanine, right? This carbon, even only this, at this positions, only in the 6th position and 7th position, there's difference, right? In the 6th position, there is a added amine group, right? And then it's, and then also at this position on carbon number, it's a 2 position, there is another amine group, right? That's the only difference. But basic structure right here, right? To synthesize, let's say adenine, guanine are coming from this amino acid, glycine, glutamine, aspartate, THF, and carbon dioxide. So you have to remember this, right? Sometimes they can just ask you, you know, to form this 2 ring, the 2 purine based structure. What are the amino acids that are actually used to make it, all right? Now, let's just say, now I'm gonna draw. Look, I'm gonna quickly make the same structure right here, okay? Here, this is a structure of N carbon, right? I'm making the same compound, HN, carbon double bond with oxygen right here, right? You have a, what is this, double bond here, this double bond, you have N, and then you double bond here, right? And then you basically N compound, right? Look, same structure, right? Same structure. Now we'll bring this guy right here, okay? And this is what? This is my, basically, this structure is what? This sugar molecule, right? So if we bring a sugar molecule right here, we'll simply say this. You have a carbon, look, you have a structure like this, right? And you have a hydroxyl group attached there, okay? And then you have a phosphate group, like this, right? When you attach, all right, this bond is a glycosidic bond, okay? Right here, on the carbon number. Now, okay, this is ribose sugar with a phosphate, okay? Because this compound, remember, it has amine group into it, right? Look at this one. There's different enzymes, a different process is going to take place, right? And we're going to make up this hypoxanthine, this compound hypoxanthine, right? When you add this hypoxanthine with this, what? Let's just forget. With this ribose sugar, we make nucleoside, right? Nucleoside. That is called, this is hypoxanthine, right? Plus this, what is this? This is a ribose sugar, right? This is what? Nucleoside, right? This is a nucleoside. Nucleoside. This compound, we call them, okay, hypoxanthine plus ribose, we call them a inosine, okay? But if you add, because we already had a phosphate group there, right? We already had this phosphate group here, so it's because we call them a IMP, okay? Inosine monophosphate, all right? This is called inosine monophosphate, okay? That's what we do. That's what we made. Now, once you make inosine monophosphate, right, what is the thing? Now we got the idea, right? How do you make the inosine monophosphate and all of them? Now, if I have to make this inosine monophosphate right here, we'll just make a simple compound right here. Carbon double bond with oxygen, right? And then you basically have a hydroxyl group here. You attach this with the different structure right here, right? Okay, I'll just write down. You got this structure, but here I'll just write down here. What is the structure I'm adding? Hypoxanthine, right? So I'll just write down hypoxanthine here, all right? I'll just write down. And then this is a phosphate group attached, right? Now, when you make IMP, it can go to two pathway. Okay, it can go to two pathway. This is something you have to remember. The pathway where you can use aspartate, all right, aspartate, or the pathway you can use a glutamine, all right? You can use a glutamine. That's the thing. Because there is an enzyme, like for example, adenocyclate synthetase enzymes are there. Like what are these enzymes? Okay, adenocyclate synthetase enzymes, right? And then here, there's another enzyme, which is called inosine, okay, monophosphate dehydrogenases enzyme. And there's the other one is called adenocyclate, right? Adenocyclate synthetase enzymes, right? Synthetase enzymes there. So, what is the point being here is that, look, in order to make, look, you saw how hypoxanthine looks like, right? If I have to make the structure, I'm going to make the structure. It's very, very important. Look at this. All right, here. Draw this compound right here, right? N compound, this is what? Nitrogen here, double bond, and you have this nitrogen like this, right? Double bond here, right? What is this group? This is a ketone group, right? And then you basically have what? You have a HN right here, right? HN. And then you have a double bond right here like this, okay? Look, I'll draw in black, right? So this is the structure, right? This is hypoxanthine, right? But hypoxanthine plus sugar molecule and the phosphate would make inosine monophosphate, right? This is a compound. Now, with this compound, right, we have to make what? We have to make either, using aspartate, you have to make AMP, or with the glutamine, we can make GMP, right? With this enzyme, adenocyclase enzyme, right? What happens? Look, you're basically doing, all right? You're basically adding, right? You're basically adding what? In this guy, if you're talking, let's just say if you're making AMP, right? Adenosine monophosphate. Let's just say you're making adenine, right? That's what we're trying to make. You're making adenine, right? In order to make adenine, with this compound, what are the changes that needs to happen? Look at the structure first here. Adenine, exactly same structure, nothing difference here, right? There's nothing much difference here too. But look at this position right here. In here, we have a ketone group attached to carbon double bond. What you need an amine group, right? You know what I mean? Amine group. So when doing that, right? The enzymes, so you need an aspartate, right? And if you remember, aspartate looks, what is it? Carbon, right? You have a carbon double bond with oxygen, you have a hydroxyl group, and then you have NH2 group here, and you basically have, what is this? Compound, ketone group right here, and then you have a H, okay, you have a hydroxyl group, if it's aspartic acid. Look at this. You're talking about this group, right? Look, basically what they do is like, there's a different process that can take place here, but ultimately is like, that's going to kick off this shit right here, right? Sorry for the language, but it's going to kick up this like double bond here, right here, right? And it's going to make a structure like this. You have a ketone group right here, all right? And then your double bond, making exact structure right here. And then you have look, this is exactly the same, right? And you have a hydrogen bond attached to it. Look, this N is here, right? Look at this. It was HN compound right here, right? HN compound is right here. Thus, when you remove this and you attach like with the adenocyclate, you put amine group. When you do this, right, you lose the one bond right here, right? And that bond is going to go down here, right? And you basically write AN, and then you basically have NH2. Look at this. This compound right here. Now, this compound plus we're going to bring our ribose sugar from here, all right? From, let's just say, ribose sugar, hydroxyl group here, you're adding our phosphate group here. You add them together, right? When you add this together, right? Now, okay, and also there's look, the carbon is also here, right? When you add, what do you make this compound? Now this compound, adenine, right? This is an adenine, ribose sugar with the one phosphate. You make adenine monophosphate, right? What are you doing? So look, you brought this like amine group from here, right? But then what happens to the rest of the compound? Those rest of the compounds will leave because there's an enzyme called adenocyclate, let's say lyase enzyme, right? So basically in this process, right, from going from this process right here, this process to this way, right, it will cleave up in the form of fumarate, all right, in the form of fumarate. So most important thing you should remember here is that, look, when you, from the IMP, which is, look, inosine monophosphate, it's the first nucleotide, okay? It is a nucleotide. From there, if you use aspartate, you can basically go and synthesize like adenine monophosphate. This is how you make it, right? Now, after this, right? After this, we have to remember, now after this, we'll come back here, right? Another pathway. That pathway is basically, look, same compound we're going to make. Same compound, nothing different. Look, you have a, all right, I'll make it blue, all right? You'll make a compound, same compound like this. And you have a nitrogen here, double bond with this one, and like this, right? And you have H group. Here you have N, correct? You have a HN, and you have a double bond here. And then this guy is, okay, again, from IMP, okay? We're making IMP. And this is a hypoxanthine we're making, and there's a ketone group here attached, right? Look, this guy, same thing, right? Look, you just do this same structure here, which is a hypoxanthine compound. Nothing change is going to happen in this compound, the ribose compound, right? You know, ribosyl phosphate is going to be exactly the same. If I use glutamine, right? If you use glutamine, now what is going to happen? Look, if I make a glutamine structure, again, carbon double bond with the oxygen, right? You have a keto, look, functional group, you have a compound like this, right? And you have a ketone group and you have amine group, right? H2N. Look, when you do this, right? What do you make? What you're basically doing is that, again, you know, there's an enzyme called basically inosine monophosphate dehydrogenase enzyme. Guys, you have to remember this enzyme. You have to remember this enzyme, right? It's very, very important enzymes. This one, you can remember adenocyclase synthase enzyme too, but I do want you to remember this inosine monophosphate dehydrogenase enzyme, okay? Because there's importance of it. Now, with this, you know, dehydrogenase, right? What are you going to do? Look, what is the difference between this one and guanine? Now, ultimately, we make with that, using glutamine, we're making what? We're trying to make GMP, right? So, look, the point is there's nothing happens in here. It's this one, it's carbon number 2 position. You need to add, in the hypoxanthine compound, if you add, let's say basically this amine group into carbon number 2 positions, right? You basically, what happens? You basically make something, you know, you make a guanine compound, right? So those, you know, they basically help to make our compound, which is helps with the adding one in the carbon number 2 position right here, H2N. All right, when you do that, this compound is basically called guanine. Okay, now this guanine, okay, you basically now add with this ribose sugar right here, all right, and then OH here, and plus phosphate. Now, what do you call them? Guanine monophosphate. Right? This is how you make adenine monophosphate and guanine monophosphate by what? By basically from the IMP, inosine monophosphate, right? Now, see, now you saw this, so we made both of this structure, right? Adenine and guanine, because this nucleotide, now when you add the polymers or chains of this, then you basically made a, what? Make a RNA, right? Ribonucleic acid. Because remember, look at the hydroxyl group. We made this hydroxyl, there's still in the carbon number 2 positions in the sugar, there's a hydroxyl group is there, right? So later, in order to make DNA, what do we have to do? We have to use this and we have to salvage it and we have to remove this, right? And we'll talk about that in the purine salvage video, how we remove this hydroxyl and how we're going to make DNA out of this. But this is just a purine synthesis pathway, right? So we're gonna only go up to here. It's very important because we'll use this to make DNA, like deoxy ones, all right? So let's do a quick recaps on the de novo of the purine synthesis. Remember, we start from a ribose 5-phosphate, right? And then we make something called PRPP, correct? And with, obviously, we also talk about this enzyme. This play a very, very important role. This is sort of like a rate limiting step of this purine synthesis, so make sure you know. And then we talk about how we synthesize IMP, and then from there we can synthesize GMP and then AMP. Or okay, up to here we have done that. Now in the next lecture, what we're going to do is that we're going to talk about how we're going to regulate, okay, regulations of this enzyme. It's very important for your exam. Okay, thank you.